Okay, in the mechanism of local anesthetics, so we must first understand how the neuronal cells conduct nerve impulses, or otherwise the transmission of information from the organ to the brain. So in general, this is the neuronal cell membrane. It has a body, it has a tail, and it is the axon part. Axon 1, 2, 3, 4, 5 axon has been shown, and it is having a myelin sheath. Is having myelin sheath 1, 2, 3, 4, 5 myelin sheath is shown here. So the nerve impulse starts from this point and gets transmitted to the next cell through this one. Okay, so first, the local anesthetic is used to treat acute or chronic pain, or to prevent the sensation of pain during procedures, generally surgical procedures or any other operations. So the first step is the conduction of action potentials in all afferent and efferent nerve fibers. Conduction of action potential from another axon here, placed here, and it is transmitting the signal to this one, and then slowly it gets transmitted. Okay. The message received by the cell is transmitted as an electrical impulse to the axon terminal. So it generates at the axon hillock and conducted by changes in the electrical potential across the membrane, across the neuronal membrane. The rate depends on the axon thickness and myelin sheath. The rate depends on the thickness and the presence or absence of myelin sheath. So it usually travels at a speed of 120 meters per second in myelin, and in 10 meters per second in unmyelinated axons. So at the rest, the neurons have a resting potential of about minus 70 millivolt. So this must be increased so that it becomes slightly depolarized. Okay. When the depolarization happens, this minus 70 becomes minus 60, minus 50, minus 40, then up to maximum of 35 millivolt, maximum of 35 millivolt, and that is said to be in the depolarized state. What causes the increase of the millivolt in the cell membrane inside the cell when compared with outside cell? It is because of the transfer of or transport of not transport, it is conduction of calcium, sodium, potassium, and chlorine. But in our local anesthetic case, we mainly concentrate on sodium and potassium. So when this is slowed down, the transport or the conduction, then the nerve will not give the impulse. Okay, so this state of depolarization to repolarization is important. So see this, this is the minus 70 is the resting potential, and action potential is plus 35. So for this minus 70 to become plus 35, we need the influx and efflux of calcium, potassium, and sodium ions. At this period, from this point of depolarization to repolarization, you cannot innervate the nerve cell to excite again. Okay, that you know. Okay, sodium channel is complicated protein with polypeptide sections, and the channel must be selective for sodium ions, able to detect and then open when the membrane is slightly depolarized, able to detect and then close when the membrane becomes hyperpolarized, then again convert to resting state at the repolarization. Okay, so at hyperpolarization, they should not open. At depolarization, they should open for the transport of ions. So this influx of ions at this depolarization state inside the cell, and this causes outside the cell repolarization that leads to resting potential. The point below the resting potential is called hyperpolarization. Okay, so the sodium ions enters to this way only, and when it is in the open state, here it is in the closed state. So you cannot have the sodium ion enter into the cell. This is outside the cell or this is inside the cell. Okay, this is the amino acid chain of polypeptide chain of this first section. Second and third is not shown. Fourth section is shown, and it is the transmembrane. Transmembrane means outside the cell, and this is inside the cell. So the mechanism is to act along the sodium channel blocking effect. When sodium channel is blocked, the conduction of impulse will not happen. So it blocks the formation and propagation of action potential. This blocks the transmitters of message of pain. The ability of the local anesthetic to block action potential depends on the ability of the drug to penetrate the tissue surrounding the targeted cell and the ability of the drug to access the binding site on the sodium channel. So it should enter the nerve cell, it should identify the nerve, protein of the sodium channel and bind, and then they should block it. That we shall see. So this is the drug entering the extracellular to the cell, nerve cell, and this is the drug, and this is converted to cation, and it is binding inside the cell and also the gate. Okay, so that is explained here. The anesthetics pass through the membrane in uncharged form. They re-equilibrate with the anionic species. Inside this part, A becomes B. This is A and this is B. B is cation. See this positive side, cation. A is uncharged. So then only it can enter. Once it is B, it cannot enter. If it is, this one cannot enter from here. Okay, so this is extracellular and this is intracellular. Now once entering and then converted to B, that is positive charged atom, either they can bind to this side or this side, from here to here, or the gate is open and this one is here. So it is access the binding site via hydrophilic pathway. This is hydrophilic pathway because inside the cell there are water, so it can become hydrophilic and then go attach here. The local anesthetic molecule is believed to bind to the binding site in its ionized form. Okay, and sometimes the hydrophobic pathway, that is where this is a lipid, so hydrophobic pathway is also followed. It depends on the drug. So tertiary nitrogen presence helps to bind with the site as hydrophobic or hydrophilic in nature. The neutral form of the drug helps to penetrate the membrane from extracellular into the intracellular. Once intracellular, either hydrophobic pathway of B or hydrophilic pathway of C is followed. Now, once it is bound, the conformation of the sodium channel gets into changed. So without drug, they will be in the open state, or inactive state or resting potential state, now closed. So these are the three states available for the sodium channel in the normal. But when anesthetics are given, one of the step is prolonged. Either inactive state is prolonged or um, resting potential state is prolonged. That depends on the drug. So there are three stages. So what is that? This is the gate. So this is closed. It may be open, it may be closed or inactive. Inactive in the case of hyperpolarization and depolarization. So when these three state is prolonged, then local anesthetics happens. So the same thing is repeated in this one. So when the membrane has been depolarized and the channel is open, local anesthetics bind. And this binding helps to change the closed and inactive conformation and thus leads to the stabilize the inactive form. Okay, so here it is been given. So small unmyelinated fibers are blocked before the larger myelinated fibers. So the which cells or which nerves are blocked first? First one is pain is blocked, then second one is sensation, then temperature. After that only this postural, touch, pressure and motor information are blocked. So the order of blocking has been given. So with this, we are concluding the mechanism of local anesthetics.